Okay, could somebody please confirm that you can hear me?
I can hear you.
I can
Thank
hear
you.
you.
Let me share my screen. Should be seeing my screen now. Today is May 16th, and we should be starting chapter 8. We're not there yet. Hopefully you started chapter 6. I don't remember if we did or not. There is no lab today, but I will be logging in for the first 15 minutes to answer any questions. So if you have a question, show up in the lab during the first 15 minutes and I'll answer your question. You do not need to show up if you do not have questions. After 15 minutes, I'll be logging out. Let me remind you, your infectious disease paper is due this Saturday at 11:59 p.m. Any questions about any of that? And you do have lab work to do besides your infectious disease paper. You also have your unknown project to work on. All right, let's see how far we got in chapter 6. It looks like we have not got very far in chapter 6, so we're behind schedule. Is this correct? I talked about the effects that oxygen has on microbial growth. Is that the last slide we talked about?
That's right.
Thank you. All right, let's talk a little bit about culture media. There is different types of media. And when you're talking about a medium, singular, it is, you use the word medium. If you're talking about two or more media, you use the word media because it's plural. Any questions about that? A culture medium can be a liquid, we call those broths, or they can be a solid. Usually a solid is made solid by agar, like an agar plate, but you could put it in a test tube and you can solidify it with other agents besides agar, like there's gelatin and starch. When you're given culture media in the lab, if you were actually to go into a microbiology lab, it would be sterile, meaning it would have no living microbes in it. An inoculum are the microbes that are introduced into the medium to get the microbes to grow. A culture is the microbes growing in or on a culture medium. There are a wide variety of media available. I used to have a little book here to kind of show you. See if I can get it. If you were in the lab, I could hold up a book. Oops, that fell out. Which is a book of the different culture media that we have in microbiology. And it's actually a little bigger than this, but it's about as thick as this. On in each page of this book, there is one culture media. So that tells you we have a wide variety of different culture media to grow microbes in. Any question about any of that? All right, no questions. Let's move on. Culture media are designed to support microbial growth. A medium must provide an energy source, a carbon source, a source of nitrogen, a source of sulfur, phosphorus, a source of trace elements, and organic growth factors if an organism needs them. Like E. coli, you would need to have an energy source, carbon source, nitrogen source, sulfur source, phosphorus, trace elements. As far as I know, E. coli does not need any organic growth factors. If we're growing something that's a photosynthetic organism, all you would need to supply, a photosynthetic autotroph, I should say, might be a nitrogen source, a sulfur source, a phosphorus source, and trace elements. And if the organism can pull nitrogen from the air, you don't even need to supply that. Any question about any of that? Agar, which is used to solidify a media, is a complex polysaccharide derived from seaweed. We usually use agar in a Petri plate or in a test tube. Agar simply solidifies the culture media to make it a solid. In tubes, we have two types. Now, let me see if I can get paint up here. In a tube, we have a deep, and that's where the agar and the media is just poured in and then it goes flat across. And then we have a slant, which will be the, oops, didn't draw that too well. Oh, well, I'm not an artist. Where the media just goes across like that. You can have a combination where, let's just draw it on this one, where you can have the slant on top of a deep, and it's usually something like that. And let me erase that now. The slant is where the auger is just put, the tube is put at an angle, and then the auger will solidify at a slant. Agar is often used in microbiology because it is not metabolized by microbes. And actually, it was Dr. Koch who first was working with solid cultures. And at first he was growing things on a potato. And you can imagine that was not a very good, I don't know, media to grow microbes on. I don't know if it was a cooked potato or a raw potato, but he was actually growing organisms on a potato and streaking them on that. And it didn't work very well. And then he started using gelatin, where he would have a plate where Jello solidified the media. And the trouble with Jello is the microbes could digest it. And then you would have this gooey liquid mess. And he didn't like that. And it was actually the wife of an assistant who suggested using agar. And agar is something you can bake or cook with. And Dr. Koch tried it and it worked very well. And as you know, we still use it today. We have different types of culture media. There are chemically defined media where the exact chemical composition is known because every molecule that is added by the person who makes the media. Like this media here only has glucose, ammonium phosphate, sodium chloride, magnesium sulfate, potassium phosphate. And if you use distilled or deionized water, H2O, okay? Meaning it has no trace elements in it. And that's all the media has. You can also have a more complex chemically defined media where you're adding a whole bunch of different molecules, like this one is adding glucose, starch, sodium acetate, sodium citrate, oxaloacetate, and then certain amino acids, organic growth factors. And obviously this would be a media that would be a fastidious organism where you have to add a lot of things for something to grow, like different amino acids. Many growth factors may be required for organisms that are fastidious, ones that require the addition of growing, adding special things to the media, like Streptococcus pyogenes, which only needs really two things, and we usually just add red blood cell, and then the red blood cell supply the two growth factors that Streptococcus pyogenes require. Normally, chemically defined media are used for autotrophs. Whoops, didn't mean to do that. Let's go back. The reason is that autotrophs don't need a lot of additives, and they can grow on most sources. An autotroph would need a carbon, actually it doesn't even need a carbon source because it can pull carbon dioxide. And if it's a chemosynthetic or photosynthetic autotroph, it doesn't even need a energy source. But if it's a heterotrophic, well, that's not an autotroph, is it? It may need an energy source if it's not a photosynthetic or chemosynthetic organism. Usually on heterotrophs, we don't use a chemically defined media. We use a complex media, and this contains extracts and digests of yeast, meat, and or plants. And these substances provide vitamins and organic growth factors that are needed by the heterotrophs. The exact chemical composition of a complex media is not known. For example, here's a complex media here, and I think this is nutrient agar. Let me blow this up. And we're putting in 3 grams of beef extract. So whatever is in the cow, whatever the cow ate, will be in the beef extract. And the exact chemicals that we're adding, we don't know. And then different batches of the beef extract will be supplying different molecules depending on what the cows were eating. Any question about any of that? Complex medias frequently contain peptone, which is just a partial protein digest. Usually you get the peptone from soybeans, but obviously beef is supplying protein as well as carbohydrates, some fat. And peptone and the beef extract supply both carbon, nitrogen, and sulfur sources. So anything that requires an organic chemical source, we usually use a complex media. Any question about any of that? All right, we have anaerobic media, and these are important for growing obligate anaerobes. These organisms must avoid exposure to oxygen, because if oxygen touches them, they will die. We usually grow obligate anaerobes. An obligate anaerobe is a anaerobe that absolutely requires anaerobic conditions. We usually do is a reducing media to grow anaerobes, and reducing media contains chemicals like sodium thioglycolate that combines with and removes the oxygen. That will result in oxygen depletion. And usually what you do when you make an anaerobic media, you pour it in a tube or something like that, and then you will autoclave it. The heating will drive off the absorbed oxygen, And then any oxygen that comes into the media will be chemically removed by the chemical. And usually, well, one is thioglycolate media. Sodium thioglycolate removes the oxygen. Any questions about any of that? We also have selective media. These are media that have a chemical that halt or inhibits a particular group of microorganisms, and it does not affect the growth of other microbes. An example of a selective media is 7% sodium chloride media. This media has 7% sodium chloride in it, which is too much salt for most organisms to grow. Most organisms do not like over 2% sodium chloride. And so 7% sodium chloride will select for a halophile, meaning an organism that likes the presence of salt. Any question about any of that? There is also differential media, and these media have chemicals in them which provide differences between different groups of organisms. The differences will not be related to how well the organism grows on the media. They usually look different, but they could have different characteristics as well, as like a different color, or an example shown here is blood agar plates. It's a differential media, and you can differentiate between the organism's ability to perform hemolysis of red blood cells. There's gamma hemolysis, which is no hemolysis, and you can see the red blood cells are red all the way up to the white microbes. There's also alpha hemolysis or partial hemolysis, where the red blood cells don't come right up to the off-white alpha. There's a ring of darkening around the alpha as well as inside the alpha. And that darkening, it usually looks dark or greenish, a greenish tint. And that's from the partial hemolysis of the red blood cells, making it look dark or greenish. And then there's beta hemolysis or complete hemolysis. And here you can see the organism in white in the B or beta. And then there's this clear region around it, as well as inside the beta. And that is from all of the red blood cells being lysed. You can actually hold this up to something and then see through, come on, where it's clear. You can actually see through this right there. And that's an example of a differential media. Any questions about that? Now, there are different types of culture media, and they can be combined, like there are selective and differential media. EMB plates, also called eosine methylene blue, are both selective and differential. EMB are selective against gram-positive bacteria, so if the bacteria can grow on the plate, it's likely to be gram-negative. EMB is also differential on the ability of the organism to ferment the sugar lactose. If the organism can ferment the sugar lactose, they'll either be looking dark, like right there, or they'll have a greenish metallic tint. And if the organism cannot ferment the sugar lactose, they'll be their normal color, which here they're looking, it looks like white or off-white. Any question about that? Another combined one is blood agar. It is both differential in the ability of cells to perform hemolysis, differential in that respect, and it is also an enriched media, so that it will allow a fastidious organism to grow. All right, there's an enriched media, that's enrich ED, which allows fastidious organisms to grow, and there's an enrichment media. An enrichment media encourages the growth of certain organisms to grow and discourages the growth of other organisms. So an enrichment media is selective, but we use it for a different purpose, and we use it to enrich for organisms of interest. It increases the numbers of desired microbes to detectable levels. It's particularly useful if the targeted microorganism is only a small proportion of the total population of cells. An enrichment media is often used when we're obtaining microorganisms from the soil or some other environmental situation or from a fecal sample. Suppose we're looking for a microbe which is causing the individual illness, like maybe gastroenteritis. Well, our feces have a ton of different microbes. And so you can use an enrichment media to enrich for the potential pathogen, which is causing the patient illness. Any question about any of that? Suppose we were interested in obtaining a phenol degrading bacteria from the soil. The soil only has a few phenol degrading bacteria and thousands of other bacteria in it. If we were to streak out a plate, hoping to see a phenol degrading bacteria, we would have to streak out over 1,000 colonies and test each one before we might get one colony that would actually degrade phenol. An easier way to do this would be to grow the soil bacteria in an enrichment media, one that is enriching for phenol degradation. How we could easily do that is if your enrichment media has only one carbon source, which is phenol, then the phenol degrading organisms will grow and other organisms will not grow because they don't have a carbon source or an energy source. Any questions about that? All right, this table is showing you the different types of media and a summary. Microbiologists usually work with pure cultures. A pure culture is a culture containing one and only one species. The reason why we work with pure cultures is if you had a test and that test were to be positive and you had 3 different microbes in the tube, you wouldn't know which microbe or maybe all of the microbes were positive and which were negative. And so, like I said, we usually work with pure cultures. How you can obtain a pure culture is get your cells from 2 colony formation events, meaning you can do a streak of a plate to get isolated colonies. And a colony can arrive from 1 or more cells. The trouble is that the first time you streak, about 30% of the colonies will be derived from 2 or more cells, in which case they may not be pure. But what you can do is take from an isolated colony and streak it on a second plate to get isolated colonies. Now, if you take cells from 1 colony, you have cells which have gone through 2 colony formation events, and those are likely to be pure. Over 95% of the time, they will be pure. Obviously, They're not pure 100% of the time. And in microbiology, it's really difficult to be 100% sure that you have a pure culture. Any questions about any of that? But if you get more than 95% of the colonies to be pure, that's good enough in microbiology, because usually we're just working with one culture, and 95% of the time is good enough most of the time. But if you only streak it out once, only 70% of the time will that culture be pure. And that's not good enough for microbiology. So to get a pure culture, you need to streak out 2 plates for isolated colony, and that's called streak plate method or streaking for colony isolation. Any questions about any of that? How you do streaking for colony isolation is you get a loop, sterilize it, cool it, and then pick up your microbes and then streak it out. Normally you do it about 20 to 30 times, so that's a little bit shy, but they're probably doing it shy so they can show you the different streaks. And you can repeat, meaning you don't have to have them perfectly separated streaks. The streaks can run over each other. Okay. And then you take your loop and you sterilize it again, cool it, and then you put it in the first streak, the first quadrant, first sector, let me put it that way, and then pick up your cells. You normally would only do it about 3 times, and this is doing it more than that. Let me see how many times they're crossing into that sector. Come on, mouse. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. You only want to go in about 3 or 4 times. What is good is that you do not want to get the culture where it is the most dense in sector 1, and that would be this region here. You want to do it where it's more dilute in sector 1, and so that's good. There won't be any organisms growing here because you haven't gone into sector 1, so I don't recommend doing it that way. What I would do is put your loop down right here in sector 2 and then immediately go into sector 1, and make sure you don't go over there. Make sure it's where sector 1 is more dilute. And then only go about 3 or 4 times in sector 1. Oops. And then streak out sector 2. And then you sterilize your loop, cool it, and then go into sector 2. You shouldn't go in as far as this, actually. Come on, mouse. You should go where sector 2 is dilute, like this region here. And then you shouldn't do it 1 time. You should do it at least 3 times. So come in and then go back and then out. Oops, did it again. And then streak out sector 3. And you might do sector 4 too. This one only has 3 sectors. And the goal is that someplace along the line, either in sector 3, 2, or 1, you'll be putting down, streaking down isolated cells, which will grow into isolated colonies. Now, in this case, it happened in sector 3, but it doesn't matter where it happens, in sector 2 or sector 1. The goal is someplace along the line, you will streak out isolated colonies, and then the colonies will grow up and be isolated. Let me blow this up. And then you can pick up from an isolated colony and streak it out again for isolated colonies. When you pick from a colony on the second plate, that's an isolated colony, the chance of that being a pure culture are very high. Okay, this is obviously a mixed culture because you can see yellow and red colonies. And if you were to pull from the yellow colonies, you would definitely need to streak on a second plate, if not even a third plate, because the first plate, we don't have isolated yellow colonies. There's no colony that's isolated that's yellow. There are some red, but even this colony here, it looks like it's only red, but let me tell you, about 30% of these colonies will give rise to yellow colonies on the second plate, meaning it looks pure, but that doesn't mean it is. Any question about any of that? All right, let's move on. Remember, microbial growth refers to the increase in cell number, not the cell size. When we're talking about growth by bacteria, bacteria grow by one of three means. Most bacteria, and I'm talking about the vast majority, over 90%, grow by binary fission. And that may be over 95%, grow by binary fission, where one cell splits into two. But a few bacteria can grow by budding, and a few bacteria can reproduce by reproductive spores. Let me talk about the reproductive spores, and then the next two slides, I'll talk about binary fission and budding. The cells that grow by reproductive spores, generally the parent cell will stand up or something like that, and it'll be a big cell. And then it'll put on the upper region of the parent cell spores. These will be little cells that will be on the upper region so that the wind or some animal coming by will pick up one of the spores and then carry the spore elsewhere, where the spore will germinate and then grow into the parent cell. And or vegetative cell, you know what I mean by parent cell. And then that parent cell can also reproduce by spores. Spores are a reproductive spore is a reproductive strategy because one cell, in this case, makes 5 cells. That is reproduction. Any question about that? And yeah, the parent cell stays alive and the 4 reproductive spores can go on and make vegetative cells. So one cell makes 5 cells in reproductive spore. So that is reproduction. Don't get that confused with an endospore, where one cell makes one cell, and then the parent cell that made the endospore dies. So one cell makes one cell. That is not reproduction. Any question about any of that? All right. Binary fission, which is what most bacteria grow by. One cell will split into two. First, they'll divide will replicate and then divide their nucleus, not the nucleus, the DNA, their genome. And then the DNA will move to opposite ends of the cell. And then the cell will literally split down the middle, where the cell will grow both a cell wall and a cell membrane down the middle of the cell, eventually dividing the cell into two. and then the cell will break apart, and then we'll have two cells from one cell. That's binary fission. Here we're actually seeing a picture of a cell going through binary fission in the electron microscope. There's the start of the cell wall and cell membrane. Any question about any of that? A few bacteria reproduce by budding, where a parent cell puts out a bud right there, and that bud will grow right there and right there until the bud will become close to the size of the parent cell, in which case it will then separate from the parent cell and become its own parent cell, where it can go through budding itself. Let's launch a little video on budding. Well, assuming it goes. There it goes. A little slow here. So we'll watch that cell there. It'll put out a bud right there. There it's putting out a bud. There's the DNA molecule, and it's dividing. And right about there, you can see the DNA molecule moving into the bud. And right about here, let's stop that. The bud is now separated from its parent cell. It's still a little smaller than the parent cell, but it'll grow. And now this cell is starting to put out a bud here, and this cell will put out a bud there. Any questions about any of that? That is budding. These are actually, that's budding in yeast, which is not bacteria, but it happens in the same way. The generation time is a concept that students actually get confused about, or often get confused about. The generation time is the time required for a cell to divide, so one cell dividing into two, and it's also the time for a population to double. So the time it takes one cell to divide into two is the same time it takes for the population size to double. The generation time varies considerably between different species. It can be as fast as 20 minutes, like E. coli has a doubling time of 20 minutes, but most bacteria have a doubling time between 1 to 3 hours. There are some exceptions, like Mycobacterium leprae has a doubling time of 10 days. So it takes 10 days for one cell to become two cells. Mycobacterium leprae is one of the slowest bacteria we know, and the Mycobacterium tend to be slow at growing because they have a lot of mycolic acid in their cell wall, and mycolic acid is a fairly difficult substance for the cell to make. So it takes the cell time to make the mycolic acid, and then the cell takes a long time to divide. Any question about it? All right. Students frequently get confused on the generation time, I think because they're thinking of always driving, where we say miles per hour. When you're talking about the generation time, it isn't units per time, it's time per doubling. Okay, so it's a little different. So when we're talking about E. coli has a generation time of 20 minutes, we actually mean E. coli has a doubling time of 20 minutes per generation. That per generation is frequently not stated. Like most bacteria have a generation time to 1 to 3 hours. It's 1 to 3 hours per generation. Any question about any of that? So don't get confused about the generation time. It's not the, what would that be? Generations per time. It's the time per generation. And there will be a quiz question on generation time. When we're talking about microorganisms, microbiologists prefer plotting them using a semi-log plot, which is where the generations is actually an arithmetic scale, the number of generations, but the number of cells is plotted on a log scale. If you did not use a semi-log plot, let me get that out of here, you would have a scale something like this, arithmetic scale, where one cell will divide into 2, 2 into 4, 4 into 8, 8 into 16, 16 into 32. You'll note that most of the points are all jammed together at the start. And then as you go up, the points take up more space on the graph. And then this next point, from this one here, the next one, it's not going to be on the graph. It's not going to be on the page. You got to put this next point on the ceiling. And that's why we do not like using an arithmetic scale. And that is all the points tend to be lumped together. And then you can't get the next one on the graph because it's going to be on the ceiling. And then this mathematical function here is very complex. We prefer plotting on a semi-log plot because right here, the points are not lumped all together there. They tend to be spread out. And I don't know what that would be because I can't count by log base 10, but you can see how these points, the ones that are shown, are pretty even. And then the next one right here isn't going to be on the ceiling. It's going to be about right there. So you can extend the graph and get the next point on. And then the real reason why microbiologists like the semi-log point plot is this function to describe the growth is very simple. It's simply y equals mx plus b. And that's why microbiologists like to plot growth on a semi-log plot. It's not a complete log plot because the generations is in the normal scale, the X scale. Any question about any of that? All right. When we're talking about growth, almost all living organisms go through different phases of growth. There are 4 growth phases of all microbes and almost all living organisms. First, when you inoculate something from a solid media to a liquid media, the population will enter a lag phase where the microbes are not growing. What they're doing is they're making the enzymes they need to grow in the new environment. And so the microbes are not growing in the lag phase. They're metabolically active, getting the enzymes they need to grow. They then enter the exponential growth phase, and initially there's lots of room around, no waste around, lots of resources around for the cells. So the cells start growing exponentially. However, as the population number increases, we will start having fewer space, more waste being built up, and there will be fewer resources around. So the growth will start to slow until we enter the stationary phase, where in the stationary phase, the population numbers are not increasing. The population number is staying stable, the stationary phase. What's happening in the stationary phase is the lucky cells are growing because they have some room around them, they have some nutrients around them, and not too many waste products around them. So they can grow. But other cells, the unlucky ones, are dying because they have very little, if any, room around them. They have lots of waste products around them, and they have few, if any, resources around them. So the unlucky cells are dying. The important part is that the cells dividing equals the number of cells dying. And that's why the population is stable while we're in the stationary phase. Okay. Eventually, though, as the population gets older, you will not have the number of dividing cells equal the number of dying cells. There will be more dying cells than dividing cells or reproducing cells. And so the population will enter the death or decline phase where the population numbers actually decrease. Early in the death and decline phase, you may have the very lucky cells actually reproducing, but late in the decline or death phase, no cell will be reproducing. You'll only have cells either staying in a non-reproductive state or they will be dying. Any question about any of that? We know this growth curve fits almost all living organisms because we've looked at it in different organisms. And one example was somebody introduced deer to an island. Initially, the deer were transported and they didn't know the island, so they didn't immediately begin reproducing. They were in a lag phase. And then they explored the island and started reproducing, and they were in exponential growth because there were lots of room and lots of resources. But as the deer population grew larger, they eventually entered the stationary phase where the lucky deer were dividing and the unlucky deer were dying. And then eventually the deer entered a decline phase because there were very more deer than the population could support, meaning the plants and the ecosystem on the island could support. And so the deer population started declining. Any question about any of that? All right, we have different ways of measuring microbial growth. There are direct methods and indirect methods. A direct method is a plate count, filtration method, the most probable number, and direct microscopic counts, where you count cells under the microscope. An indirect method uses turbidity to get the determination of how many cells you have, or you can do metabolic activity, or you can do the dry weight. All of these are indirect methods. I've used all of these methods for obtaining my cell numbers, with the exception of most probable number. I've never actually used that one to get the number of microbes present. The plate counts, also called the viable plate counts, are the most common way that microbiologists obtain the number of cells in a culture. It's the most common method. What it does is it measures the number of cells which grow into a colony, and then you make dilutions and plate out the dilutions, and then count the number of colonies to get your estimate of the number of cells present in the original solution. This usually takes more than 24 hours because it does take time for the colonies to grow on the plates, as well as time to make the dilutions and then streak out the plates. Because the number of colonies we get, we don't know if the colonies will be formed from one cell or multiple cells. So instead of saying the exact number of cells from our estimate, we will say the number of colony forming units per mil in the original solution. Is that clear to everyone? So to do a plate count, also called the viable plate count, you first get your original sample and make several dilutions of it, which is shown, I think, in the next slide, right here. Original inoculum, we can dilute it 1 to 10 by taking 1 ml of the original and put it into 9 ml of the broth. You then mix that, take 1 ml out of that, put it into 9 ml of media. Now you've gone through two 1 to 10 dilutions. And so this dilution is 1 to 100. And then you can mix that up, take 1 ml out of that, put it into 9 ml, that's the 1 to 1,000. There's the 1 to 10,000 and 1 to 100,000. You then plate out the dilutions, allow the colonies to grow, and count the number of colonies on the plate. We're looking for a plate that has between 250 and 25 colonies per mil. No, the plate will have 25 to 250 colonies, sorry. And then you say that that dilution would be 25 to 250 colony forming units per mil times the dilution of the original sample. You're getting an estimate based on your number of colonies. So you make your dilution here, and then you take each dilution and put it on a plate, which is actually shown here. You can do this by either putting in 1 ml and then moving that solution 1 ml all over the plate, allowing the plate to absorb the liquid, and then incubating the plates, usually upside down in the incubator, and then count the colonies. You could also put on a smaller volume, spread it around, allow the plate to absorb the liquid, and then incubate it in the incubator, usually upside down. In this class, we'll tell you, you always have to grow things in the incubator upside down. So some of the dilutions will give rise to too many cells to count. There's no way to count that. You can't count that one either. This one would be very difficult to count, especially if we were to count it we were to count it and base the original number on that, it'd be difficult to do because there's so many cells growing together. You can't count all the colonies. So what you want to do is let me blow this up. You want to count the number of colonies in this plate, not this one, because this one will have a mean which will be fairly good and will not range that much. Whereas this one, even plus or minus one colony, that's plus or minus 25%, a very huge range. And so we base our colony numbers in the original sample from the plate count of this plate, this dilution, and times, in this case, times this dilution, which is 1 in 10,000. So whatever that is, it looks like maybe 30 times 10,000. I can't do that in the head. 300,000. So the original inoculum would be something like 300,000 cells per mil of the original solution. Any question about that? You do not want to use this one because the mean will be very wide. Even plus or minus one colony will be plus or minus 25%. So you want to use something between 25 colonies and 250 to 300 colonies. Over 250 to 300 colonies, you can't really count the colonies. There's too many of them to count, and they'll be growing together and it'll be too difficult to count. Any questions about the viable plate count? If not, let's move on to the filtration method. It's similar to the viable plate count, except you're sampling a very low density number of cells per mil. Like, for example, if you wanted to sample how many bacteria are in your drinking water, and you were to get a loopful of drinking water and streak it on the plate, you're likely to get no colonies because our drinking water has very little bacteria in it. It's something like 10 bacteria per, I used to know that. Is it per cup, per liter, per quart, per gallon, something like that? It's not very many. And so on a loop, you're just not getting a large enough sample or inoculum to get any colonies. What you can do, though, is pass the drinking water through a sample, through a filter, and have the microbes stick to the filter, and then take this filter and put it on a petri dish, and then allow it to grow. And then you can get how many colony forming units you have per the original sample of the drinking water. And obviously you have to measure how much drinking water you sent through the filter. You notice how I said colony forming unit. In this case, it looks like all of the colonies will be forming from more than one cell. Doesn't look like there's any singlets in this picture here. Any question about that? So the filtration method is only used when the number of microbes are very dilute and you would not be able to get enough cells from streaking out a plate. There's also a direct microscopic count where a measured volume of liquid is placed under a gridded slide, and then the cells are put under a microscope and then counted under a microscope. It's not good for motile cells because they can move around. For example, here we have the gridded slide. This cell could move into the square if we're only counting this one square here. And so that would be adding one. And then this cell can move the square before you get down to it. So it doesn't work well if the cells are motile. It also doesn't work well to separate out viable cells from dead cells. For example, on this slide here, can anyone point out what's a dead cell and what's a viable cell? Is that dead or viable? We just don't know because you can't tell under the microscope. Or most of the times you cannot tell under the microscope. This is showing you the gridded slide. It does have a specific volume underneath the grid. And usually we count one square and then you multiply by 1,000,000 because this is 1 microliter of volume right here. And so we then multiply by 1,000,000 to get the number of cells per mil of the original solution, original sample. Any questions about direct counts? All right, so that's the viable plate count, the filtration method, the direct microscopic count. I'm not going to do the most probable number because I've never actually used it. Furthermore, I don't know anyone who's ever used it, so I'm not going to even discuss it. We will talk about the indirect methods, turbidity, metabolic activity, and dry weight. These methods, the indirect methods, only give you an estimate. It does not give you the exact number of cells in the original solution. You only get an estimate. Turbidity is dependent on the amount of light that gets through the media. And then you measure the amount of light that gets through the media. So that's clear media, the light shines through it, and then we measure the amount of light getting through it. If you have cells in the media, less light will get through the media. And so the turbidity will be increased and there will be less light that the turbidity meter can measure. And then you look on a chart and you see the measurement of the turbidity. And then you look on the chart for that measurement and see the corresponding number of cells per mil in the original solution. Now, initially, that chart has to be set up. So someone has to do this on many different cells or cell concentrations to set up the chart. But once you have the chart set up, you can then use it indefinitely. Because all it's dependent on is the cell blocking the light getting through. It's a very quick way. All you do is you get your tube of your cells, get your tube of blank media, put the blank media in, turbidity meter to zero it, and then you put your tube with your cells in there and see what the turbidity amount is, and then just look on the chart for how many cells you have. Any question about that? Metabolic activity looks at the metabolic activity of cells. It assumes the bacterial metabolic product accumulates in direct proportion to the number of bacteria present. In Biology 160, you used to measure the amount of CO2 that was produced. I don't know if they're still doing that, but what you did was you had cells and all you did was you measure the amount of CO2 was produced. You then look on the chart and then determine how many cells you had in your source. It does require somebody to set up a chart, but once you have the chart set up, you can use it indefinitely. Any question about that? So the more CO2 that's made in the metabolic activity, the more cells you have. The dry weight is something you can do to determine the cells. And what you do is you just pull off the liquid, dry the cells, and then at your leisure, you can go and measure the cells. The greater the weight of the cells, the more cells you have. And depending on the weight, you just look on a chart, and then it'll tell you how many cells you have. Obviously, somebody has to set up the chart the first time, but once the chart is set up, it can be used indefinitely. All right, any questions about any of that? Those are the different methods we have for counting the number of microbes. Light count, filtration, direct microscopic count, as well as most probable number is a direct method. Indirect methods being turbidity, metabolic activity, and the dry weight. Any questions about chapter 6? If not, let's finish that. And let me just see where we're supposed to be today. We're supposed to be starting chapter 8, so let's start chapter 8. Chapter 8 is on genetics. So we'll be talking about the genetics of microbes. Obviously, this slide is showing you the major goals in a rough outline. Please know the terms. We'll talk about each of them. 2, be able to describe DNA replication, RNA transcription, and protein translation. And then 3, understand the different types of mutations. This slide is showing you, we're going to talk first about the structure and function of the genetic material. We'll talk about the flow of genetic information. We'll then move on to DNA replication, RNA transcription, and protein translation. After that, we'll talk about the regulation of bacterial gene expression using operons to discuss gene expression. We'll talk about mutations. We'll talk about genetic transfer and recombination. And in this lesson, we'll end talking about genes and evolution. So genetics is the study of what genes are, how they carry information, how that information is expressed, and how genes are replicated. A gene is just a segment of DNA that encodes a functional product. It may be this segment here, or it could be this segment here, just a region of DNA encoding a functional product. That product is usually a protein, but it may also be an RNA. Any question about any of that? I think it went backwards. All right. A genome is all of the genetic information in a cell. Because our microbes, a cell is an organism, you could say the genome is all the genetic material in an organism. And usually when we're talking about human genome, we're talking about all of the genetic material within a organism. The study of genomes is called genomics. The genotype is the genes of an organism. For example, an individual with blue eyes has the genotype lowercase b, lowercase p. The phenotype is the expression of those genes. For example, if an individual has genotype lowercase b, the expression of those genes will be those individuals has blue eyes. So the phenotype is blue eyes. The genotype is the genes for that phenotype. And for most organisms, they're deep diploid. So most organisms will have two alleles or two genes per genotype. Any question about any of that? And yes, microbes can be diploid. It's especially true when the microbe is self-reproducing, tends to be diploid, having both male and female, or excuse me, not male and female. One chromosome from the mother, one chromosome from the father. The phenotype is the expression of the genes. In this case, the phenotype of this individual is brown eyes or brownish eyes. An individual that has brown eyes has the possibility of two genotypes. They can be either capital B, lowercase b, or they can be capital B, capital B. Any question about any of that? All right, this is showing you a chromosome that came out of E. coli. Each chromosome has each gene, or in this case, there's only one chromosome. So we'll have all the genes of the organism on the chromosome, which is kind of shown here. Let me blow this one up. Different genes here, here, here. And you can note that there is some matter, this right here, which is DNA, which does not code for a gene product. Eukaryotes have more non-coding DNA than they have coding DNA, meaning most of the DNA of a eukaryote is not a gene or a genetic information. It does not code for a product itself. That doesn't mean it doesn't do anything. A lot of that non-coding DNA actually helps the cell bind up its DNA to form the chromosome. So initially the DNA is unwound, not wound up, but unwound, and will be in solution of, well, this case we're talking about a eukaryote, so it'll be in solution in the nucleoplasm. But that DNA can supercoil or coil up to form a chromosome. And this happens during every mitosis and meiosis, but it does not happen usually at other times. So if you were to look at the DNA of the cell, you wouldn't see it under the light microscope because it will be a single strand of DNA, and that strand of DNA will be dissolved in the nucleoplasm or the cytoplasm if we're talking about bacteria. But in a eukaryote, that DNA may help the DNA wind up to form the chromosome. Any questions about chromosomes? All right, DNA is a polymer of 4 different nucleotides: adenine, thymine, cytosine, and guanine. You can either know these names or you can just call it A, capital A for adenine, capital T for thymine, capital C for cytosine, and a capital G for guanine. You do need to know one or the other, but you don't need to know both or whatever. DNA is a double helix with proteins inside it. By double helix, we mean DNA takes the shape of a spiral staircase. And it's a double helix because it has two strands of DNA. One strand going in this direction, one strand going in that direction. So the two strands are opposite. And then they hydrogen bond between the double stranded DNA. The sugar phosphate is the backbone of the DNA, meaning one strand of DNA is held together by the sugar phosphate covalent bonds. The two strands of DNA are only held together by hydrogen bonds. The hydrogen bonds bind two nucleotides, one on this strand, one on this strand. The A and the T hydrogen bond with each other 2 times, shown there, and the G and the C hydrogen bond 3 times with each other. The C does not hydrogen bond with the T, and the G does not hydrogen bond with an A, and vice versa. When we say the strands run in opposite directions, we mean they are anti-parallel. Any question about any of that? Let's do it here. All right, let's start talking about the flow of information within a slide. The information starts in the DNA, in the vertical flow of information within a cell, starts with DNA. That information is transcribed into RNA. So the RNA now contains the information. And then the RNA will be translated into protein. So lastly, the information can be held within the protein. The DNA, the RNA, and the proteins each have their own language via which the information is stored. The information is stored in DNA by the sequence of DNA nucleotides. The information in RNA is stored by the sequence of RNA nucleotides. The information in the proteins is stored in the sequence of amino acids. Any question about any of that? All right, I think before I go here, I think I'll talk about this next time, and that is the flow of information can flow in 3 different ways: within a cell, between generations of cells, and between cells of the same generation. Any question about any of that? If not, I'm going to stop here. If you have any questions, I'll be in the lab for the first 15 minutes, 6:30 to 6:45. After that point, I'll be logging out unless there's continual questions, in which case I'll just answer questions until the last question is answered. If you don't have a question, you do not need to show up in the lab. Remember, you should be working on your unknown project as well as your infectious disease project if you need to. The infectious disease project is due this Saturday. Any questions? All right, I'll see you next time we're in the lab.